Welcome to the talk Ribana and myself will give on the bee demonstrator in the name of several others that are mentioned here on the slide. Insects can be quite annoying, but of course they are very important, for instance, for the food web, but also for instance, the crop pollination service they provide. The global value of the crop pollination service is estimated to be about $200 billion per year. Nevertheless, several studies report that there is a loss of insect diversity and abundance. So what can farmers do to address this challenge? There are several measures, for instance, wide crop rotations, low application or no application of pesticides, organic farming, or flower plantings such as flower strips. They can all contribute to an ecological intensification of agriculture. With our demonstrator, we aim to evaluate these measures or these cropping systems, and we want to make use of low-cost microphones and cameras, as well as machine learning tools to address this challenge. In a demonstrator, we have two main objectives. The first one, Ribana will mainly focus on, is the development and application of innovative technology to detect and quantify specific pollinators in the real life in the field. And the pollinators we are focusing at are bumblebees and honeybees. The second objective is the test of a new mice-based intercropping system with flowers for the ecological intensification of agriculture. You can see here on the bottom right an image that shows maize, and in every second row we sow flower mixtures. I will now explain a bit more in detail how the experiments look like. We're conducting them since 3 years. So today, in this year, we are the third year, and the experiment takes place at the campus Altendorf. We always did some adaptations because this is an ongoing process, but we always had some key treatments, which are maize sole, so maize as you know it. Then there were treatments where we grow only Phacelia. Phacelia is the plant you can see here, which flowers very nicely in lila. You will see some images later. And then we always have some treatments where we bought or we seeded flower strips. That means we bought seeds that are usually used for floral plantings. And then we always have a mixture of them. So we have maize, and between the rows we sow Phacelia, and we have maize, and between the rows we sow the flower strips. And here it's important to know that the number of maize plants per square meter stays always the same. But in the case of the mixtures, we add then additionally some Phacelia or some flowers. That means the competition, of course, increases, the competition for light, water, nutrients, and so on. And Phacelia is extremely attractive for bees and bumblebees, but has a rather short flowering period, whereas the mixture of the flowers from these flower strip seeds, they start to flower very early. For instance, the buckwheat flowers already beginning of June, and there are some plants in it that flower very late. For instance, the sunflower, you can see it here. Or on the right side where you can see the mice that is already ripe, and we see a mallow that flowers very late in September. So the advantage of this flower strip seeds is that they flower for a long period, and thus they provide also fodder for a long period. In this experiment, we conducted several measurements. We did classical agronomic measurements such as biomass, yield, height, soil moisture, and so on. But the focus, of course, were the insects. We caught them with a net. You can imagine that it's like when you catch a fish in the water with a net. You do the same with a hoverfly or a butterfly in the air, and then you catch them and you identify them. Then this year we installed so-called malaise traps. They stay for a defined time period, for instance, 24 hours, and the insects enter, then they fly to the top because they want to disappear. And then there is this bottle where there's ethanol in it, and they die. And then you end up with this interesting soup of insects where you can then later, with this tweezer, get out the individuals and sort them and identify them. Then we also installed microphones to record the insects, and we installed cameras, action cameras. Ribana will focus on that later. I will sum up the main outcomes of the experiment now in the next 3 slides. On the right side, you can see a picture that shows nicely the experiment from this year in Campus Altendorf. On the front left side, you can see the flower seeds with the large sunflowers, and then in yellow some mustard is flowering, and in lila some Phacelia. On the right side you see the Phacelia sole flowering, and in the back left you see the maize, and in every second row there are the flowers sown in, and on the right side you see the maize, and in every maize row there is the Phacelia sown in. And in the first year in 2021, which you may remember was quite wet, there was no yield decline of maize due to the mixture, so maize was not affected by this competition. And the other 2 years, in 2020 and 2023, it was in most of the treatments affected negatively by the mixture. You can even see it here on the picture on the right side that the maize is a bit smaller, and it also has a bit rolled leaves because it was stressed, or there was a competition for water with the Phacelia. And in this year, so roughly, I mean, this year, of course, we have not yet harvested the maize, so I'm referring to biomass cuts, but in general, we can say that there's definitely a severe reduction of maize yield of up to 30 to 50%. However, there are also some treatments where there is no significant decline of maize yield, for instance, in the treatment where we have sown the flowers just every second row. Then there are some other problems like the weeding that is, of course, a bit complicated once you have sown the flowers. And then farmers are always a bit scared of seed dispersion that may be problematic because these flowers are, of course, flowering, and then they have the seeds and they fell off, and then they could be potentially weeds in the next year. So it's quite important to select the right species for this system. Now let's switch to the pollinator results, and the slide focuses on the results of the net capture. On the left side, you can see values from July 2022, and on the right side from 2021. You see here the different species. We don't have to go into detail here, of course. Here we have the Phacelia treatment. This is Phacelia with maize. Then F stands for flowers only, and FM for the mixture flowers with maize. On the right side, we have 2 dates. P again stands for Phacelia, PM for Phacelia with maize for the 2 dates. So, and first of all, I would like you to have a look at the number of insect individuals. Here they are given as individuals per 1 hour per square meter. And you can see that there is a very large range. That you can, in 1 year, in July, end of July, you could find 8 or 10, and then in the other year you found 2,000, or 900. So there is a super large variation, which shows us that that's quite critical if you have just a snapshot of insects on the one or other day, and then you, let's say, try to upscale it to a growth period or so. What you can also see here that there is, in most cases, a decline of insect individuals if the Phacelia is grown with the maize, because there are also less Phacelia plants, of course, so it makes, of course, sense. However, the decline is not so sharp, definitely less than 50%. And you can also see here, for instance, the number of insect species we identified, that they are in a similar range. So there is not a decline of species just because we added the maize. The next or last result I would like to show is the results of the visual counting. You can see here an image. The students defined a square meter of area, and then they counted all the bees or all the bumblebees in that area for a defined time. And the graph here shows the number of bees over 8 days. Yellow, the yellow box plots are the number of bees in Phacelia, and blue, the number of bees in Phacelia with maize. And you can see that there are 2 dates, here the second one, and here where there are definitely more bees in Phacelia only. However, in general, the numbers are quite similar. So in mean and average, we can say that there are still about 75% of bees in the maize Phacelia mixture as compared to sole. For bumblebees, it's a bit less, about 50%. The bumblebees seem to be a bit more disturbed by the tall maize plants. So with this, we can say that there is a high potential to increase the fodder availability and the habitats for insects with this maize-based growing system. And of course, it's also a nice playground for pollinator identification and detection. And with this, I would like to pass the microphone to Ribana.
So our main goal in this project was to develop methods that help us to get statistics about bees. And here we can distinguish between detection and tracking. So detection will answer the question, where are objects of interest in each frame? And tracking will answer the question, where are objects of interest and where do they move to? And in this project, we had a look into both and detection and tracking, and I will show you some of the results in the next few slides. Next, please. So for detection, we used a so-called YOLO V5 neural network, and without going too much into detail, what it does is it gets first an image from the field, then it extracts characteristic image features with a convolutional neural network. Then it combines these features in a very, very smart way on different levels. And then it suggests bounding boxes, where each of these boxes is assigned a confidence value, indicating whether it contains a bee or not. And the boxes with the highest confidence are hopefully the ones that really contain a bee. But such a network needs to be trained, and we also needed a good camera setup. And next, please. And this, so what we did over the time, so first we started with our smartphone just to get a good feeling about it. And then later we continued with action cams and with a specially designed camera stand, what is illustrated here in this image. It does not look fancy, but it served its purpose. So at the very end, we mounted this action cam, so we have a top view, and then with this we can record images. Next, please. So yeah, as I said, we recorded images, and to train the network, we manually mark bees in order to learn what they look like, so that the network is able to automatically detect them. And we decided for 3 classes, honeybee and bumblebee, and we had an unknown class because sometimes there were other insects, or we could not identify on our own if it's a honeybee or a bumblebee. And for the training, we used 70% of this data we collected and marked, and 15% for validation, 15% for testing and evaluation. Next, please. So, and these are our quantitative results. So the confusion matrix on the left contains absolute values and shows the confusion between the single classes. So the diagonal contains the correctly identified objects. And since we are only searching for objects, the background entry contains 0. So the right column labeled with background and with the numbers 17, 12, and 4 contains all objects which were detected but are actually background. And the bottom row labeled as background with the numbers 27, 20, and 18 contains the numbers of missed objects. So it looks quite good. And on the right, you can see the precision recall plot. And what each of these lines show are the precision and the recall for different confidence values. So precision is a measure of quality. So how many of the detected objects were actually correct. And recall is a measure of quantity. So how many of the existing objects were actually detected. And the confidence value is a threshold on the classification probability of each box. So you can see that bumblebees can be detected quite well. Honeybees not so good. There's still room for improvement, but as first results, we are satisfied. Next, please. And here you can see some quantitative results. So we trained on the full data set. Our algorithm was able to detect 1 honeybee and 2 bumblebees, and you can already see how difficult this is. And next slide, please. But here you see one challenge and one problem the algorithm still has. So we trained on the smartphone data and on one day, and we tested on the other day. And the algorithm was not able to detect a bee, although it's in there. Next, please. And very briefly, I also want to show you our results for the tracking. We used the strong sort algorithm, which is a neural network for feature extraction robust to appearance changes in connection with the Kalman filter to model the motion and estimate the position in subsequent frames. Next, please. And yeah, so here you can see a video. What you can see in the video is the algorithm is able to detect the bees and to track them, but as soon as the bees go really, really fast, you can see the bounding boxes disappear. So this is still some things we need to figure out, but overall, I think for this difficult setup, we are quite satisfied with our first results. Okay, so let's come to the conclusion. So what we want to show you, or what we show you, the question was, can AI help to quantify pollinator ecosystem services? So yes, our approach is non-invasive, so we can test it on a lot of data without hurting the bees, and it can be automated. It's at the moment limited on the poor lighting condition. We cannot record during night, of course, and windy conditions are a problem, or when insects are far away or too tiny. Some more open challenges is, first of all, we need further evaluation of a new maize-based cropping systems, and we need to transfer it to different fields in different years, different days, without new annotations. So this is something we try at the moment, and also we're still figuring out a good camera setup. The potential applications are further, for example, evaluation of cropping systems and the evaluation of management impacts on insects abundance and diversity. And with this, thanks a lot for your attention, and hope you had some fun with our nice videos. Thanks a lot. We are open for questions now, I think.